All right, now we're ready for our wonderful speaker here all the way from New York. And Dr. Tessa Pocock, did I say that right? Whew, okay. So she has her master's in plant biochemistry, a Ph.D. in plant physiology from the University of Western Ontario, Canada. She's conducted extensive research, worked in commercial greenhouses, and for a horticultural LED manufacturer in Sweden. Dr. Pocock currently works at the Smart Lighting Engineering Research Center at Rensselaer Polytechnic Institute. That's not a mouthful. And is starting a multi-disciplinary light and plant research program. Whew. All right, thank you.
I'm good to go? Okay. No, I'm not on mute, am I?
Here.
No, I'm not on. I'm good. Okay, well, thank you very much for inviting me here today. And thank you for the nice introduction, Jennifer. And one thing that I didn't include in my bio was that I started my plant science career growing cannabis in 1995. It was my first real job in the industry, and I worked for a company that had hemp stock removed from the list of illegal substances. So in Canada, we were allowed to grow fiber hemp. So my first job was to grow out 13 different strains. It's very difficult to get seeds back in those days. We had to bring them in from Hungary. And then I had to select strains that were good for growing in southwestern Ontario and also that were not containing any psychoactive ingredients. So those ones I had to cull out and get rid of. So it was my first academic publication, but now I put it on my CV. For many, many years, I didn't include that because I was always worried it would affect me getting a position somewhere. So I'm really, really happy to see the progressiveness here in the States. Okay, we're okay? Oh, do you want me? Oh, okay, I can talk louder. I'm just trying not to.
Try moving the mic. Something's not working.
Okay, how about like this? I'll just hold it. No?
Better.
Okay. Oh, we've got feedback now.
You may be right underneath the speaker.
Okay, how about this here? No? Sorry, Kyle. This is good here? Okay, I will not move from this position. So we won't have a dynamic talk today. But so I've been working with plants and obviously light for my entire career. I thought I understood light until I started to work with LEDs. As Jennifer said, I did my master's. I worked on higher plants. I worked on wheat. And for my PhD, I worked on an extremophilic algae from Antarctica and looked at how it used light, how it protected itself from light, and how on earth it could grow 17 meters below ice in a lake in Antarctica. So working with an extremophile prepared me for working with LEDs because we're seeing some really interesting things with that. Plants have a long history on Earth. They have evolved under fluctuating environments. They're sessile, they cannot move, so they have to cope with light and other environmental parameters to survive, and they've done this. So they've had to adopt an array of photoprotective mechanisms. Light is energy, it's information, but it's paradoxical. It can kill plants as well. When light energy is absorbed in excess to that utilized by the plant, it has to dial up some photoprotective mechanisms, and that's energy expensive. It's costly for the plant, so you don't want to do that. Over 100 genes are regulated by light in a plant, and 26 biochemical pathways as well. And so by understanding this, I think we can really tune into the spectrum to regulate and control the plant. And we joke at the ERC, we're all control freaks. We all work in control. And so now I want to control the plants, and we're succeeding at doing that. So I'm asked all the time, what light do plants use? And this is what we're given by industry. So does what we leave out matter? Yes, it certainly does. And not only our lab, but other labs around the world are finding this as well. And it took me a while to figure out why on earth lighting companies are giving us this kind of light to grow plants under. And I figured it out. So when you Googled, well, this is back in 2008, when you Googled LED and plants, you not only got to read through the discography of Led Zeppelin, but you found this. Well, thanks, a few old Zeppelin fans here in the crowd. So you get this. So this is an absorption spectrum. Along the y-axis, you have light absorption, and along the x-axis, you have wavelength. So this is for isolated chlorophyll. If you grind a leaf up, extract the chlorophyll in an organic solvent, this is what you get. But chlorophyll never exists like that in a plant. It's bound to one of 15 different proteins. It's embedded in a dynamic membrane called the thylakoid membrane. And when it's like this, its absorption properties change completely. So this is actually incorrect. And this is what industry has based their spectrum on. So let's look at this. This is a really nice experiment done in Vogelman's lab. They inserted a nanoprobe into a snapdragon leaf, and they measured chlorophyll fluorescence, which is a tool that I use in the lab all the time. Now, chlorophyll fluorescence is actually the re-emission of absorbed light. So by looking at chlorophyll fluorescence, you can actually estimate light absorption. It's one of the things you can estimate. You can look at light use efficiency. You can measure stress. It's a very powerful tool. And so here we have a false color transverse section of a snapdragon leaf. And you can see when this leaf, they did this work in an integrating sphere and under directed diffuse light, but we can just focus on the direct light for now. So what they found when they exposed the leaf to red light at 660 nanometers or blue light at 488 nanometers, they see that light absorption occurs at the top of the leaf. When they exposed the plant to green light, in this case 532 nanometers, the light was absorbed throughout the leaf. So if you want your whole leaf to photosynthesize, you need to give your plant a broad spectrum of light. Why would plants have evolved and wasted energy to block about 30% of the solar spectrum out? Doesn't make sense. Also, Terashima et al. Had found that red and blue light, you have higher photosynthetic efficiency with red and blue light at the top of the leaf relative to green. But when you look at photosynthesis in the lower layers of the leaf, the photosynthetic efficiency is higher under green light than either red or blue. So there's tons and tons of evidence in the scientific literature for this, which is being ignored. So even more importantly, let's look at the action spectrum for photosynthesis. The action spectrum is essentially telling you at what wavelengths work is being done. And work in this case is photosynthesis, which we measure as CO2 fixation. So we call it the quantum yield of CO2 fixation. So on the left-hand panel, you have photosynthesis based on an incident light amount, and on the right-hand side, you have photosynthesis based on actual absorbed light by the plant. In this case, it's a cucumber plant. And you can see that, indeed, photosynthesis occurs across the spectrum, and in fact, in the blue and green regions, it's very similar. So I work quite closely with the group in Cornell, the Controlled Environment Agriculture group, and we're really working on increasing efficiency of systems, especially lighting systems, in plant growth facilities. And I used to divide up CEA, as we call it, into two sectors, greenhouses and plant factories or vertical farms. But I've now included a third category, which is converted warehouses. We'll get to that in a minute. Between 72 and 94% of the sunlight in a greenhouse, or the light requirements in a greenhouse, the sun provides about 72 to 94% of it. It's free. It has a very low carbon footprint. And so greenhouses really are a good way to go if you can. However, especially in the north, we need to use supplemental lighting. And the light of choice right now is high pressure sodium. And I know that in many cases, plants have different lighting requirements during their developmental stages. And sorry, I'm right next to you here. And so that also needs to be addressed. Urban farming. I did a really great project with a company called Gotham Greens, and which are based in New York. I don't know if many of you know them. They have rooftop greenhouses. Want me to use this?
Let's try that. Yeah.
Does this work better?
I think it's on.
Can you hear me? Okay. We're good? Okay, good. Anyway, they have rooftop greenhouses, and we did some work which was funded by the New York State, nicer to New York State Energy and Research Development Authority. It's a mouthful. And so we bought 3 different commercially available LEDs, and we tested them out in the seedling area so that they would have some good evidence for purchasing fixtures for their next greenhouse, and they've got 3 now. So you can see here HPS on the left. We've got I'm not going to name names here because I'm not advertising or fixtures, but we have a blue and red fixture there on the right-hand panel. It's like an HPS analog. You can just switch them out. We've got on the bottom right there, we've got some blue and red bar lights, and at the end of the bench we have a phosphor converted blue LED fixture. So there are plant factories and vertical farms as well. And here we have one example from Japan. I met Dr. Kozai in Panama at a vertical farm conference, and he estimates that vertical farming will grow in Asia by 2,000%. It's a huge industry there, and it certainly requires a lot of lighting. On the right-hand side, you're a bit closer to home. This is Caliber Biotherapeutics. They were also at the conference on vertical farming. And they're actually producing the Ebola virus in tobacco plants. So plants provide food, material, and you can also use them as little green factories to produce pharmaceuticals or other good compounds. Converted warehouses. I've traveled extensively throughout California and Colorado. I've visited a lot of growers. We've talked about light and light requirements. And we have a long way to go. This is extremely energy intensive. They're a high light requiring plant compared to leafy greens that I'm working with now. And so we've got a lot of work to do together to make this work for everybody. This is where we're predicted to go in the future. And although these are pretty funky looking buildings, they will also require light. And in fact, the image, the rendering on the left is purported to be built in New York City, while the 2 on the right were purported to be built in Paris. Paris has this big 2050 thing going on with incorporating plants into the urban landscape. This is a really tricky slide. And so we can walk through it together. I think it's an important slide. This work was done by Cale Harbuck and Lou Albright at Cornell University. And it's not in press yet, but it will be in May. So Lou is really good at sharing information, and he shared this with me. So they've used the DOE Energy Plus to simulate a plant environment. However, they did modify it. For instance, they added plant evapotranspiration, which is an important thing to consider when you're simulating whole systems for plants. They divided it up into plant factories, which is denoted as PF, and greenhouses, which is denoted here as GH. They looked at lighting. They based the lighting efficacy on a paper that was published by Nelson and Bugbee in 2014. And there they looked at the efficiencies, although it should be efficacy, of many different fixtures. And they found that the top of the line HPS was more efficacious than any other fixture. However, the one LED fixture had the same efficacy, which is 6.12 moles per kilowatt hour. So that's an important paper to read for everybody that's interested in energy efficiency and lighting. So what do we have here? We can see right away that plant factories consume far more energy than a greenhouse. And another important thing to note here is that these simulations are based on one shelf of plants. So when you start increasing the layers of plants, you're going to increase energy consumption. There's a subdivision in the plant factories, PF1 and PF2. And PF2 is, they've inserted what they call economizer control into the model. And economizer control essentially uses and regulates CO2 addition to these facilities, as well as temperature and relative humidity. We have not started to work on lighting yet, and that's where the ERC and Cornell come into play. The greenhouse models are also subdivided. Greenhouse 1 is just a regular greenhouse. Greenhouse 2, they've inserted winter ventilation. So of course that's going to be more energy intensive. You're going to have to reheat the air as you bring it in. So plants have had a bum rap, I feel, in the world quite often, and they are certainly far from the tendency to do nothing, which is the definition of inertia. They are fast, they are responsive, they communicate, they acclimate, and they adapt. They're fascinating organisms, and I just love working in this dynamic and kinetic area. The biodiversity is immense. There's almost 400,000 species of plants on the planet. However, we're only using a small part of them. When I was an undergraduate student, I thought I wanted to be a plant hunter. And I was focusing on studying ecology, and plants around the world. I wanted to go to South America and find all of the cures for every disease, to help humanity. But I took a biochemistry course, and I got sucked in completely, and then decided to focus my career on plant biochemistry to look at what's inside those plants that makes them so interesting. So we're getting into a few technical slides here. One project that I worked on in the lab, I work in what we call sole source lighting. I don't have natural daylight in my lab. I've taken a semiconductor lab. I work in an engineering school. There's no other plant person there. I took a semiconductor lab and whatever else I could find around the department, and I built a plant lab, and it's working quite well. So this is some data from one of our trials. So we used cool white fluorescent fixtures as our in-house control. You can see this in orange on this figure. We used 2 red and blue LED fixtures. I'm calling them LED 1 and LED 2. However, I'm not sure where the pointer is on this. Is it here? Oh, yeah. So, however, if you look at the peak maximum for the 2 red and blue LEDs, there's about a 30 nanometer difference. Does this make a difference? It's red and blue. It appears the same to us. The third LED that I used, I'm calling LED 3, and it's a phosphor-converted blue LED. They're using 2 types of phosphors, which enables the spectrum to be pushed way out here, above 700 nanometers. So let's look at some plants. So here is basil. Again, everything remained the same except for the spectrum. So these are spectral responses that we're seeing. And we're seeing, for basil, good results under the cool white fluorescent. What can I say? We have, here are the 2 red and blue LEDs. And you can see the difference in the growth. The LED2 has less biomass accumulation. The leaves are a bit thinner and smaller. And here we have the phosphor-converted white LEDs. But another interesting thing here is look at the algae on the grow cube. We're seeing spectral responses of the algae in these grow systems. I use a hydroponic unit. It's really hard to kill algae in those. You kind of have to live with it, because if you kill the algae, you kill the plant. So I've got a couple of students right now, engineering students actually, I'm converting them, that are doing some growth kinetics on green algae under different spectra, so we can sort this out and try to help people minimize it using light. Here we have green lettuce. We see sort of a similar response to, as we saw in the basil. But now my favorite, the red lettuce. This is the same genus, this is the same species as the green lettuce. However, it has the capacity to produce these anthocyanins, the red pigmentation. So this is a different cultivar or a different strain. So when you think of medicinal plants and the hundreds of strains, you're going to have to really understand what strain you're growing to optimize or maximize production. So here we see that the cool white fluorescent, we have tons of anthocyanin accumulation in these plants. This is a blue and red LED here. We had good biomass, but not very good pigmentation. Here it's pretty minimal in pigmentation. Under the phosphor-converted blue, there's something in those phosphor-converted LED fixtures that plants do not like, from my experience. And we switched out LED2 because it had consistently poor performance, and we replaced it with what I'm calling LED4, which is built with clusters of red and white LEDs. And we can see we got good pigmentation here, but the leaves were really curly, and the texture, I'm a texture eater. I eat my experiments, usually. For lunch, the texture just wasn't very good. It was a bit thick, so you have to, when you're growing plants, really look at the whole plant and the quality of the plant. So I like dynamics, I like kinetics, so let's see if we can actually alter this. So let's take the plant with the lowest anthocyanin concentration and move it to the spectrum that has the highest. So what happens? It changes. So you can use the spectrum to shift, or the biochemistry of the plant. This is fantastic. So those numbers indicate hours. I set up a time-lapse camera, and we can see that by 8 hours after the shift, we see pigmentation occurring. And after 24 hours, we increase the pigmentation 17-fold. So this means that perhaps there's a place for dynamic lighting in this industry. Maybe you want to use a spectrum that is good for biomass accumulation, and then before harvest, blast it with a good anthocyanin producing wavelength. So what is that wavelength? Well, we're working on it now. Somewhere in there. And although I missed one there, actually, that's not showing up. That peak at 400 is also interesting. So I've built some fixtures in my plant lab, and I'm using a 6-chip LED that I've mounted on some good heat sinks. And we've just installed them now in the cabinets, and we started working. So we're systematically looking at the narrow bands to see what does what, so that we can then maybe replace, just add into that blue and red spectrum a couple of LEDs to produce those anthocyanins. So case closed. That study can be finished up and filed. That's what we thought. We now will understand how to increase anthocyanin biosynthesis in plants. Wrong. I decided to grow the superfood, kale. And found that under the spectrum that produced high anthocyanin in lettuce, we saw very low anthocyanin in kale. And under the blue and red spectrum, we see high anthocyanin. However, it only ever got to half as much as what is in red lettuce, so it's not really a super good source for anthocyanin, but it is for other pigments such as the carotenoids. So it's back to the drawing board with kale, and that's why when we're growing, a lot of different plants, they are regulated differently. So this is fantastic. We have one biochemical pathway that's differentially regulated by light. So why am I obsessed with anthocyanins? And I do get obsessed with things now and then until I sort them out. They're really nutritious. It's what makes red wine and blueberries healthy for us to eat. There's over 600 types of anthocyanins in nature. The one that exists, and they're divided into 6 groups, the one that is in red lettuce is the cyanidin. So I thought, okay, well maybe the anthocyanin in kale is a different type of anthocyanin. That's why it's differentially regulated. No, I searched through the literature, exactly the same. It's a cyanidin. So I have to share some biochemistry with you now. So essentially, you have CO2 assimilation, right? You've got energy. You've got carbon skeletons that can then be shuttled into growth and metabolism in the plant. And here we have the photosynthate being shuttled into a phenylpropanoid pathway, and it goes down through a series of enzymatic steps and produces anthocyanin. I'm particularly interested in the enzyme chalcone synthase. This enzyme is regulated by light, and it's the key regulatory step in this pathway. It is the the carbon skeletons, once they go through here, are committed to finish this and to go to anthocyanin biosynthesis. So this is a very important step in biochemistry, and it's light regulated. Depending on who you read, it's regulated by blue light, red light, or far red light. It's regulated by what we call photon flux density, so the intensity of the light can also increase anthocyanin biosynthesis. But what we're finding is that it's not just blue light, it's a narrow band in the blue light because the blue and red fixtures don't produce it. Or maybe it has to exist with green to be effective. These are the questions that we're going to be answering. So again, we all go back to our roots, pun intended. And why am I interested in chalcone synthase? Well, it's a member of a superfamily of enzymes called polyketide synthase 3. And this happens to be the key regulatory enzyme in cannabinoid biosynthesis. So if we can shuttle carbon flux through to the anthocyanin pathway in red lettuce, perhaps we could do the same thing in other crops whose enzymes are light regulated. Unfortunately, we can't test these hypotheses yet in the US. So here we have on the left, we have a trichome. This is, they are concentrated around the female inflorescence, and this is where the biochemistry takes place, right here. That's where all the cannabinoids, and there are over 70 of them, are being produced. And here we have the female inflorescence with a bunch of trichomes there that you see around the flower. So the energy numbers I showed you today are pretty grim. We're really working on improving them. Just at the DOE meeting, and yes, work needs to be done on green gap of LEDs, on blue droop. We can do this. Drivers control, all of that can help the efficiency of LED fixtures. However, we also have other, added value. Perhaps we should look at efficiency as microgram of anthocyanin per kilowatt hour, or microgram of medicine per kilowatt hour, instead of just looking at, watt to watt, or micromole to watt. So light regulates a number of different pathways. It's an information source. And so you have to, and you can play around with these. Flowering is important. We all know red and far red light is involved in this. Branching is important, especially for flowering crops. Plant height is really good. It'd be nice. Growth regulators are the #1 chemical used in Europe in ornamental crops, and it's to keep plants short so we can fit them on our table and be able to see the person across the way. This can be eliminated. You can modulate plant height using light alone. Of course, we have biomass, plant immunity and defense and stress tolerance. That's what I did my master's on, using light and temperature. And yes, you can increase stress tolerance, absolutely, using light. And of course, phytoceuticals. Now, a phytoceutical is defined as any compound that has biological activity. So whether it's an anthocyanin or a medicine, it's a phytoceutical. So how does this do it? How do plants do this? Well, beside plants, we're blind. They have 12 different photoreceptors that have been elucidated. And they, here you have, the newest one is a UVB photoreceptor called UVR8. We have 3 different blue photoreceptors in plants. The Zeitlube is a timekeeper. The phototropins are kind of neat. They control movement. So whether it's chloroplast movement in the cell, because they do move, it's very dynamic plants, or leaf movement, it's phototropin. Cryptochrome, ubiquitous across the 5 kingdoms, we all have it, and this actually drives biochemistry as well as some circadian rhythm entrainment. Phytochrome is the oldest known photoreceptor. There are 5 variants, and this controls in particular flowering, but pretty well everything else. Green I've included here. There's no photoreceptor that is actually being isolated and characterized. However, there are green absorbing forms of cryptochrome, and green light has also been shown to activate phytochrome. So there's all this interplay going on, and it's really a lot of fun trying to tease all this apart. Not only that, you have photosynthesis acting as a sensor and a signal, and it's the redox state of the chloroplast. When you have light absorption in excess of light utilization, you have a buildup of protons and electrons in the chloroplast that aren't being used, and this signals a cascade of events. So again, I've actually just published a review of this. If you're interested, you can check it out in Hort Science. So I can't end today without talking a little bit about biosensing. I did mention chlorophyll fluorescence. And we use that a lot in the lab to look at the physiological state of the plant. Is the plant happy? We can tell that. It speaks to us. It's also good because, well, I'll go into that later. So essentially, what is fluorescence? Well, these are the 3 fates of light energy. I don't have the 3rd one on there yet. But you have light absorption. This is photosystem 2, otherwise known as the engine of life. It's where everything happens in the chloroplast. Hope there's no PS1 people here, but I don't think so. So anyway, you have light absorption. You have this specialized chlorophyll molecule called P680. It absorbs preferentially at 680 nanometers. You have the energy state is such that it can extract an electron from water. This gets shuttled through a series of electrochemical multi-complex molecules and goes into photochemistry. That's where you want the energy to go. You want it to go into growth. However, at the same time, it's just an intrinsic property of the chlorophyll molecule. It will fluoresce about 1 to 3% of the absorbed light energy. When a plant is stressed, when you've got something happening down here, low temperature, insect infestation, drought, you will see that there will be an increase in fluorescence. It's not a protective mechanism. It's not used to dissipate energy. It's just a natural phenomenon, and it can increase up to 15% of absorbed light. So we exploit this as plant physiologists every day. Another thing that happens, just like in any system, is some energy is dissipated as heat. We call this non-photochemical quenching. We can measure this using chlorophyll fluorescence, or we can quantify this using high-performance liquid chromatography, because essentially the heat dissipation occurs because of an increase in the concentration of zeaxanthin, another plant pigment, which actually we have as well in the back of our eyes, in our macular region. It's called a macular pigment when it's in a human. We do not biosynthesize these pigments. We have to intake them through our diet. Kale is the number one source. However, as I've shown, it might depend on how it's grown and when it's harvested. Spinach is the next good source, and then after that it's about 15 micrograms per milligram fresh weight, and then you go to egg yolk, which is about 0.15 micrograms. So plant source is a really good source for these macular pigments. So we all have seen chlorophyll fluorescence. This is an image taken from a satellite. It's used to look at algal blooms around the world. And the beauty of using chlorophyll fluorescence in the lab is that you can detect changes before in a plant's physiological state before visual appearance or visual symptoms occur. When I did the lettuce shift, of course I measured chlorophyll fluorescence during that shift. I saw increases in the effective quantum yield 1 minute after I shifted the plants. And the 1 minute was because it took me that long to run from behind the laser curtain and press the button on my computer. I probably could have done it faster if I had an assistant. And so that was really fascinating. We also saw it within 1 minute a decrease in non-photochemical quenching, so indicating that there were pigment shifts occurring within 1 minute. Very fast response, and 8 hours before we could visually see it. So this is very exciting because one of the things that we're doing at the ERC is we're looking at developing a system much like Helios. There's a few people in the world actually that are doing this, 3 labs actually, one at Chalmers University of Technology, one in the University of Georgia, and one at the ERC. And so our theory is that why not have the plants control the light environment? If there are too many blue photons, we could probably sense that, and then it could turn down the blue region if it's not using it. And our motto is not a photon wasted in the lab. We want to perhaps increase efficiency through increasing light use efficiency by the plant rather than the hardware. Wished I had that a few weeks ago. I checked the plants every single day. Saturday morning, they were fine. Sunday morning I go in, pump was broken in the hydroponic tank, and this is what I saw. Now, if I'd had my biofeedback system set up, I could have had an alert saying that, there's something going on, get into the lab right away, because you would be able to sense drought very quickly as well. So when we're thinking about light and plant growth facilities, we have to take the whole ecosystem into account. And these are just a few things in the ecosystem. In the tomato house on the left, you see a bee box. They use bees to pollinate the crops. Actually, one of my summer jobs as a student was hand pollinating tomato plants with a little vibrator, which I did all day long, and flower by flower. But now they use bees. And so when the temperature is right, when the light is right, when the relative humidity is right, and it has to be right for the pollen transfer to occur, they open up the bee box and the bees fly out. When the bees are done, they go back into their box and they get closed in until the next day. Lighting is really important here. One greenhouse I visited in Finland, a tomato greenhouse, they use high-pressure sodium lamps and they illuminate 24 hours a day for pretty well 9 months of the year. One of their primary costs was the loss of bees because the bees would fly up into the HPS and fry. And so they would have to turn off the lights. Once they let the bees out, they found the flowers and they turned off the light because the bees obviously need light to move around. Also, you have to tailor the spectrum for the photoreceptors in bees for this to happen. And they are primarily, there's 5 different types of bee eye. I've looked into this a bit, and they have different photoreceptors, primarily in the green and the UV regions. Oh, sorry. Another thing, here's a nicely managed medical marijuana crop in the UK. And here they have parasitic or predator mites. that they've released into their crop to help combat or to help eat up the mites that are a pest. And this is called integrated pest management, and it's used all the time in well-managed growth facilities. Why use chemicals when you can use a biological to mop up your pest and pathogen problems? And so there again, you have to tailor the light spectrum for integrated pest management and whatever insect you're using in that. So I just wanted to leave really saying that it's not just plants that we can use. On the left-hand side there, you see cyanobacteria, tremendous amount of chromatic adaptation in this organism. And you can see a change in pigmentation just when you switch the light, and quite often temperature at the same time. But it would be interesting to look at spectral shifts in these organisms. Well, actually, there are some. Actually, it's red shifts. And here we have green algae. This is, you can see on the right-hand side, the yellow cultures. Those are cultures that are really high in carotenoids. And so you can actually alter these, again, using temperature and light, or maybe just light. So we're also working on this in the lab. So the numbers are grim for closed growing. It will get better. And even so, I'm a real proponent of locally produced foods. Plants have integrated themselves into urban spaces. They've integrated themselves into our economy. And I think that once we get the hardware kind of fixed up a bit, we can also save energy by developing appropriate spatial, spectral, and temporal light algorithms. The word light recipe is used a lot, but I feel that it kind of simplifies the case, and I feel like I should have my apron on. I prefer the word light algorithm, and Lou Albright and I coined that together because it is more than just a recipe. We're changing everything. So we can optimize photosynthesis, as I said, increase and maximize light use efficiency. That will help efficiency. We can direct plant growth and development. That's a really good sort of non-energy component. And we can regulate biochemical compounds. That's really one of my loves right now. The biofeedback system will also help reduce the energy use. And so it's really important, and it's not going to take one lab to do it. This really requires a concerted effort between labs around the world, and also is transdisciplinary. We need to get horticulture, plant physiology, engineering, architects, designers together to really make this work. And as I said, I don't have plant science students to pick from, so I've recruited biomedical engineering students at RPI, environmental engineering students, and electrical engineering students. And so I've really learned a lot from them, and I'm really happy to sort of open their eyes to the natural world. Thank you. And I can take any questions if you have any. Yes?
Thank you, John. It seems that the entire speech here was using lights to make the plants grow better. I have a project where the room sits over a wet well. It's a big room, big as this, but the whole walls are covered with algae. Is there some kind of a light bulb that'll kill the algae?
I really have to be careful of intellectual property here when I answer this question. I would actually try a light source that had about 405 nanometer spectrum, portion of the spectrum. 405. It's pretty hard to kill algae. I worked on stress in algae, and I had a heck of a time killing it. But yeah, I would try UV, perhaps, or like UVA, just a low blue. You could also maybe just try something like bleach and just physical elbow grease. It's going to be hard to get rid of, I'm sorry to say. Yeah, you're welcome. Yes, Tessa. Nice to meet another Tessa.
The biofeedback systems. All right, looking at biofeedback systems, what sort of sensors can be used that are simple, practical for people in the field to measure the biofeedback systems using different, maybe light spectrums, beyond measuring yield or lab tests after the plant is dead? What sort of simple, practical measurements can we take while the plants are growing?
There aren't any right now. And actually, a student in my lab, and we presented a poster at the DOE last night, has built a homemade sensor, and then we compared it to my $25,000 pulse amplitude modulated fluorometer that I bought from Heinz Walz in Germany. So we are looking at trying to develop some low-cost sensors. We can't do as much we're just using a photodiode filter, lock-in amplifier, which we need to get rid of because that's expensive. So we're looking at that, and then we need to actually apply functionality to a very simple signal. And so that's what we're doing, and we found some very good correlations, just measuring changes in what we call steady state fluorescence and comparing that to more complicated measurements, and we're seeing a very good correlation. So hopefully we'll, finish developing that, and it will come to market. But it's not ready yet, I'm sorry. So that question was another sensing question, obviously. Okay, I guess that's it.